Welcome to the Deep Dive, the place where we transform complex technical source material into actionable, thoroughly understandable knowledge. If you're looking for the ultimate shortcut to being truly well-informed, well, you've definitely found it. And if you value this kind of deep dive analysis, do us a favor and make sure you follow this deep dive right now. That way you'll never miss another essential insight into the forces shaping our world. Today we are tackling what feels like the ultimate genetic paradox. Modern genetics promised us this clear roadmap to health, a binary output almost, good or bad, risk or safety. But for millions of people who undergo comprehensive testing, the reality is much less clear. They encounter this informational black hole right there in their DNA report, a finding labeled something like a unique mutational change, or more clinically, a variant of uncertain significance, a VUS. It really is the definition of ambiguous data. It's a finding that is somehow simultaneously harmless and potentially harmful, all until proven otherwise.
And that ambiguity, that uncertainty, is the engine driving tremendous patient anxiety. It's a huge issue. So our mission today is really to cut through that complexity. We are diving deep into the VUS, using the material you sourced to help distinguish the scientific noise from the actual signal. We want to help manage the resulting emotional fallout and, maybe most critically, provide you with a clear, evidence-based action plan for long-term vigilance. See, as genetic sequencing gets faster and cheaper, these VUS findings are actually becoming the new standard outcome of thorough testing. So understanding this is crucial for anyone interacting with modern medicine today.
Absolutely. So let's start right at the beginning. We need to define this genetic gray zone properly.
Okay, so the current flood, and it really is a flood, of VUS findings is a direct consequence of this extraordinary technological leap we've seen in genomics. For decades, researchers were limited. They could maybe test single genes, often manually. It was slow work. But then came high-throughput sequencing, what most people know as next-generation sequencing, or NGS. This lets us peer into the human genome with just unprecedented speed and depth. We can sequence entire exomes, that's the protein coding part of the genome, or even whole genomes, quickly and increasingly affordably.
Right, and the speed is just incredible. But it sounds like it's outpacing our ability to actually understand everything we're suddenly seeing.
That's exactly it. This technological acceleration has created an enormous informational gap. The technology generates these vast amounts of data. It identifies known, validated disease-causing changes, sure, but also literally countless individual genetic differences that maybe have never been documented before. So a VUS is simply one of those identified changes, maybe a single letter swapped in the billions of letters of DNA where the clinical significance is currently unknown. By definition, it's neither definitively harmful nor benign. The core challenge is that we, as scientists and clinicians, can't yet reliably tell just by looking at the sequence which changes are harmless variations and which might actually impair how a cell functions.
Okay, that makes sense, but it still seems a bit counterintuitive.
Right.
These findings are often labeled rare variants, yet you're saying they're incredibly common on genetic reports. How can they be both rare and common?
Yeah, that's a great question, and it confuses a lot of people. They are common on reports precisely because each individual VUS is typically exceptionally rare within the general population. Think about it like this. When you test, say, 20,000 genes in one person using NGS, you're bound to identify variants that might only exist in that single person, or maybe in just a handful of people globally. Now, if a variant is only found in 1 or 2 people in the entire world, well, we have basically zero population data on it. We likely have no functional studies, no results from family members to help us classify it definitively. It's this acute deficit of information that forces the lab to label it a VUS.
Ah, okay. So the rarity is why it's uncertain.
Exactly. Our sources frame this perfectly. The rapid advancement of sequencing technology has accelerated the detection of novel and rare variants far, far beyond the capacity of researchers to fully interpret their biological consequences. This leads directly to what clinicians are now calling the variant interpretation crisis.
Wow. And I guess that crisis gets even worse when you consider de novo variants, right? Those ones that are brand new in the patient, not inherited from either parent.
Precisely. If the change only exists in the patient and wasn't inherited, you lose even the context of family history to help track it. It becomes a total knowledge vacuum for those unique changes, often locking it into VUS status for potentially longer.
Okay. So this really underscores the need to reassure people, doesn't it? That finding a VUS isn't necessarily abnormal.
Absolutely. That's why we have to emphasize the fundamental nature of our DNA. Discovering a VUS is actually a normal, even expected outcome of doing extensive genetic testing these days. Genetic difference is just intrinsic to human individuality.
We often talk about DNA like it's this perfectly standard operating manual, but it's really not. It's full of personalized deviations, little changes. Every single person possesses thousands of DNA changes that deviate from what we call the standard human genetic code. And the vast majority of these differences, thankfully, have absolutely no discernible effect on how our cells function. They're just the genetic equivalent of having unique fingerprints or snowflakes. It's actually quite amazing to think about every single person listening right now has thousands of these unique identifiers. The more genes you test, the more of these unique, unclassified differences, and thus more VUS results, you're inevitably going to find.
So if finding these variations is essentially normal, why does getting a VUS result cause such immediate alarm for so many people?
Well, it often comes down to two major misconceptions. One is fueled by language, and the other by sort of outdated scientific thinking. Let's tackle the language first. The word mutation. Yeah, I wanted to ask about that. It feels like such a loaded term. If, as we'll discuss later, something like 91% of these VUS eventually turn out to be benign, why do we stick with a word like mutation that just seems designed to cause panic?
It's highly problematic, I agree. In the public sphere, the term mutation carries these intensely negative, often frankly terrifying connotations. It leads people to incorrectly associate any detected alteration with abnormality or inevitable disease. Studies confirm this. People hear mutation and immediately think something is badly wrong, fueling that fear of an unavoidable health crisis.
Right. It implies the blueprint itself is fundamentally flawed. But the reality check is that the vast majority of genetic variations, including most VUS findings, are entirely harmless. The system often just defaults to the most technically precise language, even if it's scary. We have the example of BRCA1, right? Everyone has the BRCA1 gene. It's a critical tumor suppressor. The increased cancer risk only arises when an individual carries a specific variant in BRCA1 that actually impairs its function. A tiny unclassified change in that gene that doesn't impact its function is just a spelling difference, effectively. It's not necessarily a fault.
Exactly. And the second high misconception is this lingering idea of one gene, one trait, a kind of genetic oversimplification. This belief suggests that a single gene directly dictates a specific health outcome, completely disregarding the incredibly complex interplay between multiple genes, environmental factors, lifestyle choices, all the things that actually govern most common health conditions. Most chronic diseases, like heart disease or type 2 diabetes, are polygenic, many genes involved, and multifactorial. Believing one VUS dictates your destiny is falling prey to this oversimplification. And unfortunately, that only serves to exacerbate anxiety and can push people toward unnecessary medical action.
Okay, that sets the stage really well. We understand the VUS arises from powerful tech finding rare changes we don't yet understand, and that common misconceptions fuel the anxiety. So to try and bring some necessary order to this potential chaos, the American College of Medical Genetics and Genomics, usually called the ACMG, developed a really critical framework. If you're going through genetic testing, or especially if you've received a VUS report, understanding this 5-tier classification system is, well, it's basically mandatory. It's the standardization tool used across all clinical laboratories pretty much globally now.
It is. This framework provides a much-needed structure for dealing with that inherent uncertainty we've been talking about. We can categorize the 5 tiers pretty clearly, moving from high certainty of harm down to high certainty of being harmless. So first at the top, you have pathogenic. That's tier 1. This means there is overwhelming scientific evidence indicating the variant causes or significantly contributes to disease risk. We're talking typically greater than 99% certainty here. These are generally actionable findings. They guide medical decisions pretty much immediately.
Okay, 99% certainty. That's clear.
Then tier 2 is likely pathogenic. Here, there's substantial evidence suggesting disease causation usually follows somewhere between 90% and 99% certainty. These are also typically considered actionable, although sometimes a clinician might want a secondary confirmation step before, say, recommending a major intervention.
Makes sense? Then we hit the big one.
Right. Tier 3, variant of uncertain significance, or VUS. This designation means there is simply insufficient evidence right now to confidently classify the variant as either pathogenic or benign. The uncertainty here is the widest. It can range anywhere from, say, 10% up to 90% certainty. It's that massive gray zone.
So it's really stuck in the middle.
Exactly. Then we move towards the more reassuring end of the spectrum. Tier 4 is likely benign. This means there's strong evidence suggesting the variant is harmless, with less than 10% certainty of it causing disease. And finally, Tier 5 is benign. Here, there's overwhelming evidence that the variant does not cause or contribute to disease, so less than 1% certainty of any harm.
Okay, that spectrum is helpful. And the really crucial point about the VUS, that tier 3, is its clinical ambiguity, right? It cannot confirm or rule out a specific diagnosis if you already have symptoms, nor can it reliably predict your future disease risk if you're currently asymptomatic. It's purely an observational finding, just waiting for more data. It's like, yeah, like you said, an information gap.
Precisely. And even within that broad VUS category itself, there's actually internal heterogeneity. It's not quite a single uniform status. The category is broad, right? It encompasses variants that might have some suggestive evidence pointing toward either extreme, but not enough to make the call. So labs might informally categorize variants as, say, VUS high. This might mean the computational prediction tools, or maybe the initial patient data suggested it leans a bit towards the pathogenic side. Maybe it falls in a critical part of the gene's code, for instance. These are often prioritized for deeper investigation down the line.
And conversely, I suppose there are VUS low variants.
Exactly. VUS low suggests there's some initial evidence pointing towards it being likely benign. Maybe it's located in a non-critical region of the gene, or perhaps it's been seen rarely in healthy people before, but just not enough times or with enough certainty to fully classify it as benign yet.
But neither of those informal labels are actually actionable clinically.
Correct. Neither VUS high nor VUS low is considered technically actionable under the official clinical guidelines, but they do provide important context, especially psychologically. If you receive a VUS low classification, it might offer a degree of psychological relief knowing the lab isn't immediately worried. If it's VUS high, that might warrant prioritizing it for things like long-term family-based variant evaluation, which we'll talk about.
Okay. We spent a lot of time establishing the uncertainty and the anxiety this generates, but now, here is the statistical fact that you really need to hear, the one that should immediately help mitigate that fear response when you first see VUS on your report.
Yes, this is probably the single most important data point for you, the listener, to internalize right now. The statistical trend is overwhelmingly positive. When VUS results are eventually reclassified, and this happens over time as more scientific evidence accumulates, the data shows that approximately 91% are ultimately downgraded to either benign or likely benign.
Let that sink in. 9 out of 10 times, that VUS that's causing so much worry is going to resolve favorably. It'll turn out to be harmless variation. That fact is absolutely fundamental for patient counseling, isn't it?
It is. This statistical reality helps frame the VUS not as some impending threat, but more accurately as a temporary information gap that will, in all likelihood, resolve harmlessly. Only about 9%, the remaining minority, are eventually upgraded to pathogenic or likely pathogenic status. Knowing this percentage allows you, the patient, to hopefully gain the necessary psychological distance to resist that unnecessary anxiety, and it prevents people from immediately jumping into what we call the sick role before the scientific evidence actually warrants it.
That 91% figure is incredibly powerful.
Okay,
so we have the framework, we have the statistical reassurance, but where the test came from matters hugely, right?
Yes,
Yeah.
critically. Despite that strong statistical reassurance we just discussed, the reliability of any specific VUS finding depends massively on the origin and the methodology of the test that was performed. Not all VUS findings carry the same clinical weight. Determining the accuracy of the result itself is really the essential first step after you get that report. We absolutely have to make a mandatory distinction here between the two primary testing sources.
Okay, let's define the gold standard first. Formal clinical testing. What does that involve?
So these are tests ordered by a qualified healthcare provider, a doctor, a genetic counselor. They're usually administered in a clinical setting, like a hospital or a specialized clinic. And crucially, they utilize sophisticated, high-quality methodologies like next-generation sequencing, NGS. These methods typically sequence the entire gene or the entire coding region of interest, looking for essentially all possible changes, whether they're rare or common. And maybe most importantly, these labs operate under rigorous external accreditations. Specifically, they need to be CLIA and CAP certified.
Right, CLIA and CAP. Can you briefly explain what those mean? Because they sound important.
They are the quality control seal, essentially. CLIA stands for the Clinical Laboratory Improvement Amendments. These are federal regulatory standards here in the U.S. that ensure lab test results are accurate, reliable, and timely. CAP, the College of American Pathologists, provides an additional voluntary accreditation program that reinforces even higher standards for quality control, accuracy, and lab practices. So if a lab is KLIA-KK certified, you can be highly confident that the technical finding itself, the fact that the DNA change exists, is reliable.
Okay, so that's the clinical gold standard.
Yeah.
Now contrast that with the other main source,
Yeah.
direct-to-consumer or DTC testing.
Right. DTC tests are marketed directly to the public. You buy them online or in a store. They often use less comprehensive technology, typically something called single nucleotide polymorphism, or SNP, chip arrays. These SNP chips don't sequence the whole gene. They only analyze a pre-selected subset of known variants or common hotspots across the genome. They're just checking certain specific positions that are known to vary commonly in the population. And importantly, they usually offer pretty limited support, often just online FAQs or maybe basic reports, leaving consumers sometimes struggling to interpret complex results. This is especially true when people download their raw data and try to interpret it using third-party online services, which often use outdated or unvalidated methods.
And this difference in technology is massive, isn't it? It completely changes how you should view a VUS result from a DTC test.
It absolutely does. It fundamentally changes the risk profile of the VUS. A VUS reported by a CLIA-certified clinical lab is almost certainly a validated biological finding in your DNA. A VUS reported by a DTC lab, especially from raw data analysis, well, it's highly suspect and might not even exist biologically. It could just be a technical error.
Why such a big difference? Why are the DTC results so much less reliable for these rare VUS findings?
It comes down to the technological limitation of those SNP chip arrays used by most DTC companies. They perform poorly when it comes to accurately detecting very rare genetic variants, which, as we've established, is exactly what a VUS tends to be. SNP chips are primarily designed and validated for detecting common variants found in many people. They often lack the specificity, the sensitivity, and the coverage required to accurately identify rare, potentially unique changes. This leads to a surprisingly high number of technical errors, what we call false positive, for rare variants.
So the tool just isn't really designed for the job of finding rare variants accurately.
Pretty much. In fact, to underscore that limitation, technical artifacts, these false positives, can actually outnumber true positive results across all the rare variants assayed when using SNP chips in some studies. You are often statistically more likely to receive a technical error report than a genuine biological finding for a rare variant on these consumer platforms.
And we have some hard statistics to back this up, don't we? And they are pretty alarming.
They are. Clinical studies that specifically looked at this issue found that 40% of potential mutations identified by DTC services in genes known to be clinically important were simply false positives when they were later retested using comprehensive clinical sequencing.
40%. 4 out of 10 findings were just technical errors, not real biological changes.
Exactly, and that number gets even worse, dramatically worse, for specific high-risk genes that people worry about, like the BRCA genes associated with hereditary breast and ovarian cancer. For individuals who are not of Ashkenazi Jewish ancestry, studies reported the false positivity rate for disease-causing BRCA1 and BRCA2 variants reported by DTC services was between 69.0% and a staggering 89.7%.
Wow, up to nearly 90% false positive. That's incredible.
It is. So if you get an elevated risk signal or a VUS from a DTC test, particularly for a rare variant, you absolutely must assume, based on this data, that it has a very high probability of being a technical artifact, not a real finding.
And even for the variants that are technically real, that the DTC test correctly identified, there's still a risk of misinterpretation, right?
Yes. Misclassification is also rampant. Variants that DT providers might flag as having increased risk are often later reevaluated by clinical labs using ACMG guidelines and found to be benign, likely benign, or indeed VUS. This leads to tremendous confusion and, of course, unnecessary worry for consumers.
Okay, so this leads to a pretty clear mandate then.
It does. Any VUS result derived from DTC testing, and especially anything coming from interpreting raw data using third-party tools which might rely on outdated or unregulated databases, must be treated with the utmost skepticism. Extreme skepticism.
So if you have a VUS from a DTC test, the absolute required action is mandatory confirmation. Is that right?
Mandatory confirmation. You must go through a healthcare provider, get a referral for confirmatory testing of that specific variant by a CLIA-certified clinical laboratory. This is the only way to know if the finding is real. because, as the data shows, that DTC VUS has such a high probability of just being a technical blip rather than a genuine biological finding that needs clinical follow-up. Clinical testing remains the gold standard because of those rigorous KELI-ASAC quality controls that minimize the risk of these technical errors leading to false VUS findings.
That distinction is absolutely critical. Verify the source before you worry.
Okay, so we've established the statistics, that 91% likely benign trend, and the absolute need for source validation, especially with DTC tests. Now, we really need to address the profound human impact of living with this ambiguity. The inconclusive nature of a VUS creates significant psychological distress. There's no way around that. Receiving a VUS result often places individuals in this state of informational limbo, and studies show this directly translates into anxiety, distress, and what's called decisional conflict that can unfortunately persist for years while waiting for answers.
And this ambiguity, this limbo, it often pushes the individual into what sociologists call the sick role, doesn't it? They start believing they have some kind of impending yet undefined illness. This can lead to chronic worry, and critically, it often drives increased and potentially unnecessary utilization of healthcare services.
Yes, the sources really highlight these behavioral consequences. Patients have reported actively changing their health behaviors, sometimes drastically, and pursuing unnecessary medical procedures. For example, insisting on repeated, expensive cardiac testing based solely on finding a VUS in a gene vaguely related to heart conditions, even when their general measures of distress might seem outwardly manageable. They end up spending significant time, money, and emotional energy trying to manage a risk that the scientific community hasn't even proven exists yet.
Wait, so just to be clear, patients are actively spending money undergoing potentially invasive tests based on data that the scientific community explicitly says they should ignore for clinical decisions. Can you give us another concrete example of that kind of unnecessary testing?
Oh, it happens quite frequently, unfortunately, particularly when a VUS is detected in a gene that's associated with a severe condition that people understandably fear, like hereditary cancer predisposition syndromes or cardiac arrhythmia risks. We have documented cases where patients have insisted on much more aggressive or frequent surveillance scans than guidelines recommend. Some have even seriously discussed prophylactic surgery like removing breasts or ovaries based entirely on a VUS, finding that, remember, has a 91% chance of ultimately being benign. They often misperceive the risk, even after counseling. They tend to overestimate the significance of the VUS dramatically, and that misperception profoundly influences their decisions, sometimes pushing them towards really radical medical procedures prematurely.
That feedback loop between the ambiguity and the anxiety sounds really difficult. And it seems like it might be compounded if their regular doctor isn't fully equipped to handle these results either.
That's a major contributing factor. Data shows that many non-genetic specialists think your primary care physician, maybe a general cardiologist or oncologist, often feel unsure about how to correctly interpret and explain a VUS report, especially to a patient who lacks a clear family history of the related disease. They themselves are often uncertain about the true clinical implications, and sometimes they might inadvertently validate the patient's anxiety because they aren't confident pushing back against it.
So this knowledge gap among general practitioners can actually make the patient's misperception worse.
It absolutely can, and that's precisely why the formal clinical guidelines are so incredibly strict regarding taking action based on a VUS. Okay, let's really unpack this. This is probably the most critical clinical guideline we'll discuss today. Because of that psychological toll, the high risk of misperception and the danger of over-intervention, current professional guidelines from the ACMG and all major medical specialty colleges are explicit. VUS findings are not considered clinically actionable. They should not be used to alter a patient's medical management. That means no changes to surveillance schedules, no changes to screening frequency, and no changes to therapeutic decisions based solely on a VUS.
This is the non-actionable rule. We really can't stress this enough, can we? Ignoring this guidance risks significant harm. We're talking about unnecessary invasive procedures with their own risks, repeated expensive testing draining resources, exposure to unnecessary radiation from maybe years of extra screening, plus the inherent financial costs and the physical toll of all those actions, and just the time and resources required to analyze and manage these VUS results also puts a strain on healthcare systems globally.
Absolutely. The path forward, according to all guidelines, is clear. Clinical management must be based entirely on the patient's existing personal health factors and their documented family health history, not solely on the inconclusive VUS result. Your doctor should be treating the patient, managing their known risks, not treating the VUS. The VUS should simply be logged as a data point that needs to be tracked over time, awaiting reclassification.
And that really brings us full circle to the critical role of genetic counseling in navigating all of this, since many non-genetic specialists struggle with explaining a VUS correctly.
Referral to specialized genetic counseling professionals becomes absolutely essential. They are trained specifically to bridge this knowledge gap. Counseling provides that personalized risk assessment, digging into the family history properly. It offers the necessary emotional support and, crucially, the context needed. A good counselor can help shift the patient from being a passive recipient of confusing data to becoming an active participant in an evidence-based management plan. They can clearly explain the low statistical risk associated with most VUSs, help identify if family studies are a viable option for resolution, and firmly reinforce that clinical management should remain focused on established risk factors, not on this ambiguous finding. They are really the essential guide through this informational minefield.
So getting that expert guidance is key to managing both the psychological impact and preventing unnecessary medical steps. Okay, so a VUS isn't necessarily a permanent label. It's often a temporary status, right? The ultimate goal is for that variant to eventually be classified as either definitively benign or definitively pathogenic. And that process is called variant reclassification.
That's right. Variant reclassification is the standardized ongoing process where the pathogenicity assignment, that 5-tier classification of a genetic alteration, gets updated as new clinical or functional data accumulates over time.
What drives that reclassification? What kind of new data helps?
It's driven by several key factors accumulating in the scientific community. We rely heavily on large-scale genomic databases that pool information from thousands, even millions of people. Improved bioinformatics tools, those computational prediction algorithms, get better over time. And something called deep phenotyping is crucial.
Deep phenotyping, what's that?
That means carefully correlating the detailed symptoms, clinical observations, and health history of patients with their specific genetic findings. So if eventually 1,000 people worldwide who all share the exact same rare VUS, also all present with a very specific set of severe symptoms, the evidence for that VUS being pathogenic grows much stronger, even if we don't fully understand the biological mechanism yet.
Okay, so it's about gathering more evidence over time. What's a typical timeline for this resolution? How long might someone have to wait?
That's the million-dollar question, and the answer is it's highly variable. It can range anywhere from just a few months, if decisive evidence emerges quickly, to many years or even decades for some variants. And frankly, some unique variants, especially those de novo changes we mentioned, the ones brand new in a person, might actually never be definitively classified simply due to a persistent lack of sufficient data. So patients really must understand that resolution is often a long-term process. This emphasizes the need for that long-term vigilance and follow-up we keep mentioning.
But there is some responsibility on the lab side too, isn't there? They don't just issue the report and forget about it.
No, definitely not for clinical labs. Clinical laboratories have both ethical and often contractual responsibilities for tracking variants they've reported. They maintain proactive reclassification policies, especially for changes that were initially uncertain but might become actionable if upgraded. So, if a VUS is later upgraded to a pathogenic or a likely pathogenic result, the testing laboratory is responsible for issuing a revised report. And critically, they need to ensure that the ordering clinician is notified so they can recontact the patient to discuss the new implications. This is a really important safety net in the system.
That's good to know. But it sounds like the patient can't just be passive in this process either.
Absolutely not. You, the patient, must actively partner with your healthcare providers. You need to ensure you have a clear tracking plan in place and understand how you will be notified of updates, even if those updates might arrive years down the road. Don't assume you'll automatically hear "stay engaged."
Okay. So, besides waiting for the scientific community, are there more proactive ways to help resolve a VUS? You mentioned family studies.
Yes. One of the most powerful and often readily accessible methods for potentially resolving a VUS is through family segregation studies, often abbreviated as FSS. This is a targeted evaluation. It involves testing specific biological relatives, parents, siblings, children, sometimes aunts, uncles, or affected cousins to see if they also carry the exact same VUS that was found in the initial patient. The main objective is to see if the VUS tracks or co-segregates with the relevant health condition within the family pedigree. Does it consistently show up in affected family members and stay absent in unaffected ones?
And this is often how you get that definitive evidence needed for reclassification.
It can be incredibly powerful evidence, yes. For example, imagine you have a family with a clear history of a condition that follows an autosomal dominant inheritance pattern, meaning just one copy of the altered gene is enough to cause symptoms. If that specific VUS is found to be present in multiple relatives who have the condition, but it's consistently absent in all the relatives who don't have the condition, that provides very strong evidence arguing for pathogenicity. Conversely, if that same VUS is found in numerous healthy family members across multiple generations, that provides strong evidence arguing for a benign classification.
That makes a lot of sense. But testing multiple family members sounds like it could get expensive quickly. Is that a barrier?
It certainly could be, but here is a significant practical advantage that everyone with a VUS should absolutely ask their provider or genetic counselor about. Many major commercial clinical testing laboratories now offer dedicated VUS resolution programs or family studies programs. Under these programs, they will often provide the necessary targeted testing for the specific VUS in eligible biological relatives completely at no charge to the family.
Really? Free testing for relatives?
Yes, often free. They do this because resolving VUSs benefits everyone, the patient, the family, the lab itself, by improving its database, and the broader scientific community. It's a way to eliminate the financial barrier and give patients a direct, accessible mechanism to generate the critical data needed for variant resolution. So definitely ask if your VUS qualifies for one of these programs.
That's a fantastic tip. Okay, so beyond family studies, what about the cutting-edge science? Where is the long-term resolution heading?
Well, we currently rely quite heavily on those computational prediction tools to try and guess what effect a variant might have on the protein. But as we noted, we have to highlight their imperfection. Performance assessments show wide variability between different tools, and some studies found that the least reliable methods incorrectly predict that more than 1/3 of known benign variants are actually disease-causing. So this inherent variability and inaccuracy in prediction is a major reason why so many variants get stuck with the VUS designation in the first place.
So the computational tools aren't the final answer.
Not yet, no. The real long-term solution lies more in direct experimental validation. Specifically, there's a lot of excitement around emerging high-throughput experimental methods. One key example is multiplex assays of variant effect, or MEEs.
MAEs, multiplex assays.
Okay.
What do they do?
Think of them as super-fast biological testing systems conducted at a massive scale. Researchers can use relatively simple cellular models like yeast or bacteria or sometimes human cell lines. They can engineer these cells to contain the human gene of interest, then systematically create versions of that gene containing every single possible VUS, sometimes thousands of different variants at once. Then, they use clever biological assays to rapidly measure how each specific variant actually affects the protein's function in that system.
Wow. So instead of painstakingly testing variants one by one in the lab over potentially decades, they can test thousands, maybe even all possible variants in a gene almost simultaneously.
Precisely. That's the power of multiplexing. MAVs generate large-scale functional data for potentially all possible variants within a disease-relevant gene. This functional data can then be combined with clinical information and sophisticated machine learning models to create highly accurate lookup tables for variant effects. This is fundamentally how the science is trying to race ahead to catch up with the sequencing technology. It's about systematically addressing the massive scale of this interpretation problem, which involves potentially billions of possible variants across the entire human genome. MAVs offer a path to pre-classify variants before they're even found in patients.
That sounds like a game changer for the future. But in the meantime, while the labs are working on MADs and maybe family studies are underway, is there anything patients themselves can do to track the status of their VUS?
Yes, absolutely. Patients can proactively monitor the evolving scientific consensus regarding their specific VUS by utilizing the excellent public database maintained by the NIH called ClinVar.
ClinVar. Okay, how does that work?
ClinVar aggregates genetic variant information and its relationship to human health from hundreds of sources worldwide. This includes clinical testing laboratories, research projects, expert review panels like Klingon, and literature databases. It offers a centralized resource for variant classifications. You, or more likely your genetic counselor helping you, can search ClinVar using the standard gene symbol like BRCA1 or MLH1, or even better, using the specific technical string of letters and numbers that precisely names the variant's location and change. This is called the HGVS expression, and it should be listed on your clinical test report.
Okay, so you can look up your specific variant. Are there any pitfalls or caveats people need to be aware of when using ClinVar?
Yes, a few important ones. First, you need to understand the concept of aggregation and the potential for discordance. ClinVar displays an aggregate record which summarizes all the submissions it has received for that particular variant. You must be prepared for the possibility of seeing conflicting classification situations where different clinical laboratories or researchers have submitted different interpretations. For example, one calls it VUS, another calls it likely benign.
Why would there be conflicting results?
This discordance often stems from older submissions made before the standardized ACMG classification guidelines were fully developed and adopted, particularly submissions made before around 2014. Different labs might have used different evidence or criteria back then, so if you see conflicting results in ClinVar, it's generally wise to prioritize the consensus opinion, if one exists, or classifications provided by well-established expert panels like the clinician consortium groups, over submissions from individual labs that haven't undergone that expert review. And of course, always remember the official NIH disclaimer for ClinVar. The information is intended for research and informational purposes. It is not intended for direct diagnostic use or for making medical decisions without thorough review and interpretation by a qualified genetics professional, like your genetic counselor or physician. It's a tool, not a replacement for expert advice.
Excellent points. ClinVar is a powerful resource, but needs careful interpretation. Okay, we've covered a huge amount of ground. The ambiguity of the VUS, the critical importance of the testing source, the psychological risks, the non-actionable rule, and the long-term path towards resolution through reclassification, family studies, and new science like MBEs. Now let's try to consolidate all of this into a concrete, step-by-step action plan for anyone who receives a VUS result. What are the absolute priorities? Priority 1, consult a genetic counselor. We keep coming back to this, but it really is the single most critical step. You need that personalized risk assessment, help managing the inevitable anxiety, and guidance in establishing a sensible long-term follow-up plan. They provide the necessary emotional support and clinical context that the raw report itself completely lacks. They can help you filter your personal and family health history through the lens of that ambiguous finding and determine the actual next steps, if any.
Absolutely number one. Then, if and only if your VUS originated from a direct-to-consumer DTC test, the second mandatory action is verification. You must request clinical-grade confirmation of that specific variant from a KELA-CAP certified laboratory through a healthcare provider. You have to definitively rule out the high possibility of it being a technical false positive result. Do not skip this step based on consumer data. You simply cannot rely on it for VUS findings.
Right. Verify if it came from DTC.
Yeah.
And the third key action.
Vigilance. Long-term vigilance. You, the patient, need to be proactive. You should plan to follow up perhaps annually with your provider or genetic counselor, specifically to ask if the classification status of your VUS has changed.
Annually. Why so often if changes can take years?
Because reclassification happens whenever new research drops or enough data accumulates. An annual check-in ensures the loop stays closed. It confirms that the laboratory's reclassification tracking system is still linked to you through your provider, and it keeps the VUS on everyone's radar. Given that resolution can take years, even decades, maintaining that link proactively is crucial. Don't just assume you'll be contacted years later. Check in.
Okay, counselor, verify if DTC. Vigilance. That's a clear action plan. Now let's shift gears slightly and address a critical non-clinical area. Legal protections. What rights do people have regarding their genetic information, especially these uncertain VUS results?
This is really important. In the United States, there's a key federal law called the Genetic Information Non-Discrimination Act, usually known as GINA, which was passed in 2008. GINA provides essential legal safeguards against the misuse of genetic information, and that explicitly includes VUS results and family medical history.
Okay, GINA. What does it cover specifically?
GINA primarily covers two main areas: health insurance and employment. First, health insurance. GINA makes it illegal for most health plans, including group plans, individual market plans, Medicare supplemental plans, to use your genetic information to make decisions about your eligibility, your coverage, or, importantly, your premiums. They cannot charge you more for health insurance based on your genetic code or family history. It also prohibits them from imposing pre-existing condition exclusions based on genetic information.
That's a huge protection for health insurance. What about employment?
Second, employment. GINA makes it unlawful for employers, generally those with 15 or more employees, to use genetic information in decisions about hiring, firing, promotion, pay, or any other terms of employment. Employers are also required to maintain the confidentiality of any genetic information they might possess about an employee and must store it securely, separate from regular personnel files. This protection is vital. It safeguards you, even if a genetic variant hasn't yet led to the manifestation of any disease symptoms.
Okay, so Gina sounds pretty comprehensive for health insurance and jobs. But there's a but coming, isn't there? Are there limitations?
Yes, and this is the critical GINA limitation that everyone absolutely needs to internalize. GINA does not apply universally. Specifically, it does not regulate the use of genetic information by companies offering certain types of private insurance. The main gaps are life insurance, disability insurance, and long-term care insurance. These types of insurance are generally regulated at the state level, and protections vary widely and are often much weaker than GINA's federal protections.
Ah, okay. So those specific private insurers could potentially ask about or use genetic information, including a VUS, when deciding on coverage or setting premiums.
They potentially could, yes. It depends on state law and company policy. This is a major gap in the federal protection. Therefore, a patient who is considering applying for these specific types of private insurance, life, disability, long-term care, should exercise extreme caution. They should strongly consider consulting with a knowledgeable financial advisor who understands these nuances. In fact, many advisors strategically recommend that individuals delay undergoing clinical genetic testing, or at least delay formally receiving official reports containing things like VUS results, until after those specific private insurance policies are fully secured and in place. The rationale is that the existence of a known VUS, or even just a documented family history suggestive of risk, could theoretically impact your premiums or even your eligibility for coverage in these currently unregulated sectors. Timing can be really critical here.
That is crucial advice. Secure those specific policies before potentially creating a genetic record that could be used against you in those contexts. Okay, one last area. Data privacy risks, particularly for those who might have used DTC services and maybe share their data.
Yes, this has become an increasingly complex issue. The massive growth of consumer genetic databases has created significant privacy challenges, and the data generated has implications not just for the individual who tested, but potentially for all of their close biological relatives who share DNA.
What's the main risk people should be aware of?
One significant risk is access by law enforcement. Agencies in the U.S. and increasingly elsewhere are utilizing both public and private genetic databases, including those derived from DTC consumers who opt into sharing or upload data elsewhere for forensic investigations trying to identify suspects or victims through familial DNA matching. This practice is possible because these databases contain uniquely identifying detailed genetic information.
And the highest risk comes from sharing raw data.
Generally, yes. The highest risk to your genetic privacy likely occurs when users download their raw genetic data file from a DTC provider, like Ancestry DNA or 23andMe, and then upload it to publicly accessible third-party databases, often those promising additional health or ancestry interpretation, like GED match, for example. When data is submitted to these more open public platforms, it often becomes accessible to law enforcement under their terms of service or via legal processes. So you absolutely must weigh the perceived benefit of getting potentially unconfirmed, maybe even inaccurate analysis from some of these third-party services against the very significant and generally permanent loss of genetic privacy, not just for yourself, but also for your biological family members who share segments of your DNA. It's a serious trade-off.
Definitely something to consider very carefully before uploading that data anywhere else.
Okay,
Let's bring this deep dive home. Can we recap the essential, actionable recommendations one more time? Hashtag outro.
absolutely. Let's distill it down to 5 key takeaways. First, maintain clinical detachment. A VUS finding, on its own, should not guide your medical treatment or surveillance plans. Remember that powerful statistic. 91% of reclassified VUS findings are ultimately downgraded to benign or likely benign. Trust the process. Let the science catch up.
Second, verify the source. If your VUS came from a direct-to-consumer test, mandatory clinical-grade confirmation via a CALIC-SM-FACAP certified lab is absolutely required to rule out a technical false positive before you worry further.
Third, prioritize expert counseling. Immediately consult a board-certified genetic counselor. They are essential to contextualize the VUS within your personal and family history, to help manage the anxiety effectively, and to prevent decisional conflict based on uncertainty.
Fourth, pursue resolution actively. Don't just wait passively. Inquire about and, if eligible, utilize those often no-cost family segregation studies offered by many clinical labs, and establish a clear plan with your provider for long-term tracking of potential reclassifications over the years or decades it might take.
And finally, fifth, Be aware of legal limits. Understand that while GINA provides strong protections for health insurance and employment discrimination, you need to exercise extreme caution and perhaps consult a financial advisor regarding the potential use of VUS reports when applying for private life, disability, or long-term care insurance. Secure those policies first if possible.
That's a fantastic summary. The VUS really does represent this fascinating, challenging intersection in precision medicine where our human science is still racing hard to catch up with the sheer power of our sequencing technology. It's an unavoidable artifact of that progress, isn't it?
It truly is. And perhaps the final thought for listeners is this. The primary action when faced with a VUS is often simply to pause. Pause, seek that expert professional context, and embrace what's really a shared responsibility model. The laboratory and the scientific community track the evolving science behind the variant. But you, the patient, in partnership with your clinical team, must ensure you remain linked to that system, checking in periodically so you actually receive the reclassification update when or if it eventually comes, even if that update is years or decades away. That's how we transform this uncertainty into actionable clarity over the long run.
Pause, get context, stay linked. Excellent final advice. Thank you for diving deep into the complex VUS gray zone with us today. If you found this deep dive valuable, if it provided clarity or useful actions, and you want more of these essential shortcuts to knowledge on complex topics, please make sure you hit that subscribe or follow button right now. Join us for the next deep dive.